Hey everyone, welcome back. In our last video, we did the hard work. We took raw FASTA sequences, calculated their properties like GC content and length, and neatly packed them into a Pandas data frame. But numbers in a table are not enough for a research paper or even a presentation or any kind of research. You need visuals to understand better or explain better. So earlier in this series, we used Matplotlib, if you might remember, to plot a genome trace. Matplotlib is powerful, but making it look good takes a lot of code. So today I'm introducing you to Seaborn, a library built on top of Matplotlib, specifically designed to make uh, gorgeous publication-ready statistical plots with almost zero effort. So let's jump into it right now. First of all, make sure all these libraries are installed in your environment, virtual environment. So you can install them. You might know the code to install that. I'm not going to go into that part again. And then let's set up our data set. We are going to simulate a real bioinformatics scenario. Let's pretend that we sequenced 500 genes from a traditional crop variety, right? And 500 genes from a genetically improved variety, and we want to see if their GC content or sequence lengths differ. Okay, so for that, we will first do it like this: generating dummy genomic dataset. Okay, uh, 500 improved and 500 traditional, right? Then for that, traditional might have slightly lower GC and shorter lengths. Okay, just assume that. Okay, we are not going to be exactly biological about this, so we're just going to go based on assumption, right? So np.random, this is for creating a random data set for us so that you can follow me along and you won't have to download any external data set for this. Size is equal to 500, right? That should work. Now we are just going to copy this and paste it again, and this one is for length, right? So for that, uh, let's go with 1200. Scale will be much higher, let's say 200, and size will be 500 again. Then next, we will create uh, like improve, right? So improved might have slightly higher GC and longer lengths. Okay, so for that, we will go with suppose 46. Scale we can go with 2.5, and size will be 500. And similarly, we will go for length of improved. We will go with 1500. Scale will be 150, let's say, and size will be 500. Okay, this will create our data set for us, and now we will convert it into a Pandas data frame. So let's just run this for now. Okay, and then we can run pd dot data frame. Let's say variety will be categories. Then let's call it GC content. GC content will be np dot concatenate, and we will concatenate the uh, GC traditional and GC impute, not imputed, sorry, improve. Okay, right. And finally, sequence length np dot concatenate, we will go with length of traditional and length of improved, right? That should do it. Now let us display the few rows of our dataset. Let's run this and okay, I think I'm missing something over here. A comma is required over here. I think that should work. Okay, so run this and we have a beautiful data frame representing 1000 extracted sequences ready for analysis, as you can see here. Okay, now maybe we can check the size as well. So, right, so we have 3000 items in our list, I believe. Maybe not. One second. Yeah, sorry, 1003 rows, right? Okay. Now, the suppose the PI, principal investigator, or your superior or someone else asks you, is the GC content significantly different between the traditional and improved varieties? So the absolute best way is to show this with a box plot, right? So with Seaborn, a professional box plot is literally one line of code. So yeah, we'll see how it goes, right? So for that, we will use sns.set theme for setting the theme of our plot. These are just customization code, right? This does not count as plotting the graph. So, but still we are going with that. So I can tell you some more things about how to basically customize your plot, right? So yeah, palette will be set to, let's go with set to. Then we will create the box plot, plt dot figure. I think that f will be smaller. I don't know. Okay, yeah. Then figure size will be how much should we go for? Let's go with 8 and 6. Okay. Then sns dot box plot data will be data frame, obviously. X will be uh, variety, right? We use variety for that. Yes. And Y will be GC. Sorry, I think GC content. Yeah, spelling should be correct. Make sure. Let's do some more customization. This is not required, but this is for uh, better viewing your plot and make it more presentable, right? So GC content comparison, traditional versus varieties, and what else? What else? Yeah, we will add font size. I think that's the correct command. Hopefully, we will see. And font weight will be bold. Right, that should do about the title of the plot. Now we will label the x and y axis. Plt dot x label, that will be uh, crop variety, right? And let's go with font size 12. Similarly, we will do the y label, and y label will be GC content. We will add a percent, and font size will be 12, right? Okay, no need to save it for now, so we will just show the plot, right? Let's see what happens here. Uh, okay, I think we are having a spelling mistake somewhere. Yes, this we have accidentally said as GC content. And okay, now it should be fine. Yeah, okay. Okay, and look at that. Seaborn automatically colored the boxes, calculated the medians, mapped the quartiles, and even plotted the outliers as little diamonds or circles. Right. So from this plot, we can instantly and visually confirm that the improved variety has a higher median GC content. Right. So if you want to save this as well, I'll give you one more code. For that, you will have to do plt.save figure. Then you can name the figure as well. GC, what should we call? Boxplot dot png. Then DPI will be 300. That is the standard, I guess. And UX inches will be tight. Okay, I think that should work. And yeah, here you have your plot saved for you, right? Okay, next, what if we want to look at the two variables at the same time? Let's see if there is a relationship between sequence length and GC content. And let's color code the dots based on the variety. Okay, so for that, we will create a scatter plot with a hue. So for that, we will do uh, figure, mm, figure size. We can go with 10:6 this time since we will have even more things to show, right? So now we will do scatter plot. Inside the scatter plot, we will use data is equal to df. Then x will be sequence. Let's not make any spelling mistakes this time. And y will be GC content. U will be variety. This magically color codes the dots for us. Okay, and alpha is another property that we can add here is equal to 0.7. This will make the dots slightly transparent. Okay, here we have the plot settings, and now similarly we can just copy these things for making it easy for us. Right, so some things we can change here is sequence, yeah, sequence length was this, GC content, font size 14, and font weight for bold we can leave it like that. Now for the x-axis we will have to rename this. We will rename it as sequence length in base pairs, and this will be GC content only, and it will be same, right? Now we can save it as something else. We will name it length versus GC scatter. It's good to name your plots exactly what they are. Do not try to keep shortcuts. That can get complicated later, right? So let's try and run this, and yeah, okay, something is there. Yeah, we misspelled hue. Okay, now let's run again, and look at that. By simply adding hue is equal to variety, Seaborn did all the heavy lifting of splitting the data, assigning colors, and building a legend for us as well. We can clearly see two distinct clusters forming, hinting at the biological differences between these two groups. Okay, data visualization doesn't have to be painful. By combining pandas and Seaborn, you can generate journal-ready figures in minutes, not even hours. So remember to always use plt.save figure with a high DPI when you are ready to submit your paper. I think DPI 300 is the industry standard, but you can check the requirements and based on that you can save your plot. So we have built up an incredible pipeline now, from reading faster files to building data frames to visualizing the distinct cluster of our data. Right. So, but what if we want the computer to automatically learn how to classify these sequences for us? So, in the next video, we are crossing the threshold and we will be moving towards machine learning, and we will be looking into the libraries that involve machine learning and other things. And the code will be available for you in GitHub as always. And if Seaborn just became your new favorite library, please hit that like button, subscribe, and let me know in the comments what kind of data you will be plotting using this library. So I'll see you in the next video.